Okay, hello, welcome back to virology. We are going to make some virus particles now. We have talked about making proteins and nucleic acids, so let's put it all together and do some assembly of particles. If you look at a virus particle, if you look at the structure, you should be able to figure out how it's made. At least that's the goal for today. And virus particles come in three different general types: icosahedral, with or without an envelope, enveloped helical, and complex. And today you'll be able to look at how each of these varied virus particles are built. All virus particles undergo a common set of assembly reactions. You start out, of course, by making the structural units. Remember, a structural unit is made up of subunits. Viral protein is the polypeptide chain is the subunit. And then you make a shell by interaction among the proteins. The shell could be a nucleocapsid, RNA protein, DNA protein, or it can be an icosahedral shell. And then, of course, you have to get the nucleic acid in. We're going to talk about that today. How do you get viral nucleic acid in without putting in cellular nucleic acid? That would be a waste to have virus particles with cellular nucleic acid. So we have to have what we call selective packaging. And then there's a bifurcation here. Some viruses, of course, acquire an envelope, a membrane, and then they're released from the host cell. As other viruses do not acquire a membrane, and they simply mature in the cell and are released from the cell. So we'll see examples of both of those pathways today. Of course, just like almost every other part of viral reproduction, we depend on cellular machinery. And there are many aspects of assembly that require cellular machinery. We need chaperones. These are not people who make sure you get home early from a date. They are proteins in a cell that make sure other proteins are properly folded. Right? So when proteins are made, they can have mistakes. They can misfold. Chaperones check them out and make sure they're folded properly. And those cellular chaperones do that for viral proteins. Of course, there are also viral chaperones that fulfill the same function. We have transport systems, of course, to move components throughout the cell. We have the secretory pathway, which brings glycoproteins from the endoplasmic reticulum to the plasma membrane and out. We have nuclear import and export machinery and many other pathways as well. These are just some of them. We'll touch on these today when we talk about assembly of particles. And one of the considerations, of course, is something we brought up a long time ago, is that the cell is a crowded place. Things don't just diffuse in a cell. They are motored around within the cell. And so we talk about short distance movements. At the top of this slide, we've talked about, for example, viruses crossing membranes, the plasma membrane or some other membrane in the cell. If it's a nuclear membrane, we talked about how components can get through the nuclear pores. These are short distance movements. And then there are long distance movements, micrometers to meters even. Meters. What cell would be a meter long? Can you think of one? A neuron, yeah. You can have meter-long neurons. So viruses can get in peripherally at neurons and travel all the way up to the brain. So they can go a long way. And that kind of long-distance movement is on motor proteins and cytoskeletal tracts, for sure, and especially in those long ones. Now, here on the right is an experiment that shows you the importance of cytoskeletal frameworks for movement of proteins. This is a cell infected with vesicular stomatitis virus, where the nucleocapsid encases the viral RNA in the cytoplasm, and then eventually it's packaged into virus particles. And the movement of that ribonucleoprotein, as we'll see later, depends on motor proteins. So this is a cell stained with DAPI. You can see the nucleus is blue. And then the microtubules are stained with an antibody to tubulin, which is a major component of microtubules. You can see they're crisscrossing throughout the cell. Major cytoskeletal component of the cell, and along which many components move. Showed you movies of that during virus entry before. And in green is showing you the viral nucleocapsid protein. We're using an antibody to the nucleocapsid protein and then a fluorophore that fluoresces green. So that's nucleocapsid. You can see it is throughout the cytoplasm. It's near the nucleus all the way out to the plasma membrane. Now on the bottom, we have treated the infected cell with a drug that disrupts microtubules. You can see they're no longer nice filaments, right? They're little speckles on the bottom there. The drug is called nacatazole, by the way. It's not important, but it disrupts microtubule function. And now you see the viral nucleocapsid is hung up in very specific places in the cytoplasm, in big depots, if you will. And they're yellow because they're mostly co-localizing with the red. So the green plus the red gives you the yellow color. So they're not moving where they should be because the function of the microtubules have been disrupted. So this is just an example of how important it is to be able to use these pathways to move around. All right, so movement on motors is very important for assembly. And so everything I'm going to tell you today about movement, just remember it's happening on some kind of motor protein on a microtubule. Now, the other key point is that nothing happens fast in dilute solutions. And so viruses typically concentrate all the components in very specific places to get around that, to increase the concentration of what's needed. And we saw that briefly for RNA synthesis. Most plus-strand RNA viruses reproduce their genomes on the surface of vesicles rather than having them float around in the cytoplasm and having to run into the components they need. They're on the surface of vesicles. And these components that are concentrated, we can often see them by light microscopy because they're so big. They're much bigger than the virus particles. And we call them factories or inclusions because they were described years ago before people understood what they were. So they said, it's a virus factory. So there are two examples here. One is, as I just mentioned, poliovirus infected cell. The RNA synthesis is done on the surface of these double membrane vesicles. So in this cell on the lower left, we have a poliovirus infected cell. And you see a lot of dark dots. Those are newly synthesized virus particles. So this is maybe 6 hours into infection. And all of the endoplasmic reticulum and the Golgi is all gone. You have to take my word because you probably can't tell. It's all gone. And instead it's been repurposed to form these double membrane vesicles. And on the surface of those is where viral RNA synthesis occurs. And then, of course, since the viral RNA is there, that's where the newly synthesized virions are. They're taking up the RNA right around those vesicles, so they're near them. And then the example on the right is rabies virus. And these are called Negri bodies after an Italian pathologist of many years ago who saw these in the spinal cord, probably of someone who died of rabies, and named them after himself. And they are, this is a staining of a virus-infected neuron, actually. You can tell it's a neuron, has a roughly pyramidal shape. This neuron has within it a number of these dark blobs. Those are not virus particles. They're way too big to be a virus particle. They're Negri bodies. Those are the places, those are the factories where rabies virus is being assembled. By the way, this neuron is sick. You can tell because it's got spaces between it and the rest of the tissue. It's not supposed to be spaces there. It's supposed to be all solid. It's dying, and that's part of what rabies does to you. Now, viral proteins need to go to the right places. They have addresses to do so. All kinds of addresses are known and have been studied, and they're listed here. We need to get viruses and viral proteins to membranes, so they're membrane targeting sequences. There are signal sequences which tell the protein to get translated into the ER lumen, of course. There are also fatty acid modifications on proteins that target them to membranes. We'll talk about those. There are signals to stay in the membrane. So some proteins need to stay there, not leave. Membrane retention. There are nuclear localization signals, of course, to get into the nucleus. And there are signals to get out, nuclear export signals as well. And on the bottom is just an example of a nuclear localization signal. Can be quite simple. Here is one in T antigen of SV40. So proteins are all made in the cytoplasm, right? No exceptions. They're all synthesized there because that's where the ribosomes are. And then many of them need to get in a nucleus, so they need an NLS. And SV40T goes in the nucleus. to kickstart DNA synthesis. And it's really a very simple series of amino acids, as you can see there. Hydrophobic, 3 to 7 basic amino acids and another hydrophobic. And you can take that and put it in any protein and it will go in the nucleus. It's recognized by the import machinery. And the bottom is just a longer form. There's some more complicated MLSs, signals. So here's an overview of proteins getting in the nucleus of various viruses. So there's your typical cell with an uncrowded cytoplasm, right? It's not really realistic. Here's our nucleus, endoplasmic reticulum, Golgi apparatus. Influenza virus, the nucleoprotein goes in the nucleus so that it coats the RNA. The RNA is reproduced in the nucleus, so it has an NLS, it gets into the nucleus. Parvovirus, capsid proteins, the virion proteins need to be made in the cytoplasm, get in the nucleus where the DNA is being replicated. The same with the adenovirus structural proteins, the same with the polyomavirus structural proteins. They're all made in the cytosol. They go in the nucleus in order to encase the viral DNA, and they have to have nuclear localization signals in order to do that. And they go through the nuclear pores. These proteins are small enough that they can be transported directly through the pores. Now, in this slide, we're showing proteins moving to the plasma membrane. It's another route of transport. Proteins that are destined, say, for the plasma membrane. They're initially made by ribosomes that are bound to the endoplasmic reticulum, and the protein is translated into the lumen of the endoplasmic reticulum. So, normally they're ribosomes floating around the cytoplasm, and they're making proteins that are staying in the cytoplasm. But these target the endoplasmic reticulum. They have an N-terminal signal sequence, which targets them for the ER. And then there's a transport pathway that will take those proteins, wrap them into vesicles, and those vesicles will then be transported to the cell surface on microtubules, as you can see here. But they go through the Golgi apparatus. They fuse with the Golgi. The proteins are now in the Golgi, and then they move by vesicle transport through the Golgi, the various stacks, as we call them, of the Golgi. Eventually, the vesicles pinch off, go up and fuse at the plasma membrane, and there's our protein in the plasma membrane. So vesicular transport pathway is what I'm describing here for getting proteins from the ER to the plasma membrane. Yes? Do viruses encode their own motor proteins? I am not aware of any. So I won't say no. I will say I'm not aware of any, because there may be some that no one has discovered, but I wouldn't be surprised if they might, right, to make it more efficient for their own cargo. Yeah. Okay, now let's talk about assembly. First concept, subassemblies. Think of an assembly line for making an automobile. When I was a kid, that was an exciting thing. I don't know about you guys nowadays, but assembly lines were invented, by Henry Ford, and instead of building a car on its own, you put it on a conveyor belt and everybody added parts. It was just a revolutionary thing. Well, viruses have been doing that for years. They do assembly lines, basically. And here is the building of a bacteriophage, showing you subassemblies. So you build, for example, the phage has an icosahedral head, it has a tail, and it has tail fibers. And so you build the head, the tail, and the tail fibers on separate assembly lines. You build the tail here, and you start with all these numbers are just individual proteins that are made and put together. You put the base plate together, you put this shaft in the middle, and then you put the discs around it. And now you have a tail. At the same time, you're building the icosahedral capsid, you put that on, and then you build the tail fibers, and you put those on. So that's what happens in many virus assembly pathways. You make intermediates. And why do we do this? Well, because it makes sure you do things in the right order. Right? You don't put something on when it should be on later. And if something goes wrong, you can reverse it. So if you make a tail that's not quite right, it's not going to fit onto the head, and therefore it's thrown away. And chaperones can assure that. So quality control is a big reason for doing this kind of subassembly. We see this in a lot of virus assembly pathways. And here are 3 strategies for making subassemblies. In other words, the way you get to a subassembly can vary. So you can make it, as we saw in the previous slide, from individual protein molecules. right? So we had all of the tail proteins, for example, being made and assembled into a tail. So that's one strategy, assembly from individual protein molecules. So we're looking at the pentamer of SV40, which is the subunit of the capsid. 5 copies of VP1. The VP1 proteins are made simply by translation on ribosomes, and they fold, and then they assemble with each other. All that information for them to fold properly and assemble into a pentamer, all that's built into the amino acid sequence. So individual protein molecule. Another strategy, which we see in many virus-infected cells, is to make what we call a polyprotein precursor. That may not be a word that you're used to using, but virologists use it all the time because many viruses make polyproteins, which simply means you have several individual proteins in a precursor that then needs to be cut by proteases. It always amazes me how few people even know the word protease. When I say the protein is cleaved, they have no idea what I'm talking about. And the reason I'm saying this is Paxlovid is an inhibitor of a viral protease. And people don't get that. You really have to explain it to them. Interesting, right? Because I've thought about this my whole life, and it's natural, so you have to be very careful. You have to explain what you mean by cleavage. I think people think of a meat cleaver, right, when you say cleavage or other things. So protein precursor is the case for poliovirus, the capsid structural unit, which is made of 3 subunits, VP1, 2, and 3. That's made as a precursor. You can see VP1, VP3, and VP2. They're made as a single protein. That protein then folds up, and then a viral protease called 3CD-Pro cuts it, cuts those loops joining the wedges that are stacked together. It's a viral protease. And if you wanted to, you could make an inhibitor of that, but they tried that years ago, and you got resistance too easily, so none of them were made. Yes? So in the polio precursor, the capsid subunit, right, the structural unit, which is made up of VP1, 2, 3, and 4, that's made as a precursor. So they're all joined, they're made, they come off the ribosome all in one, they fold, and again, we don't have any cleavage yet, and then here, 3CD-Pro, that's where the cleavage happens, okay? So that's the precursor. Call it P1, actually. All right, so those are 2 ways to make subassemblies. And then, of course, added to the mix is that some proteins need a chaperone in order to fold properly. So here we have the hexon of adenovirus, which is a trimer of a single protein, protein 2, which is made on ribosomes as a single protein, folds up, and then a viral chaperone, this happens to be a viral chaperone, helps it to assemble into the trimer. Without the chaperone, the trimers don't form. All right, so chaperone is another aspect of it. Here's another couple of examples of chaperones, and these happen to be cellular chaperones. Again, you don't need to know the details here. I want you to understand the concept that either viral or cellular chaperones can help viral proteins to fold. So at the top we have the production of the retrovirus capsid, which we're going to see in some detail later today. It starts with a GAG precursor, and many of these assemble to form a shell. And there's a cellular chaperone called TRIC, T-R-I-C, which helps GAG fold into the proper formation in order to undergo this series of reactions. And then at the bottom, the polyomavirus incorporation of the pentamer into the virus particle. So here's the pentamer, 5 copies of VP1, and you can see there are 5 of those around the 5-fold axis of symmetry. Those being incorporated into the particle require a cellular chaperone called HSC70. It also requires energy. And look what other protein is in there required for building the capsid, large T antigen. It's just an amazing protein, does so many things, needed for assembly. So, and then taking this even further, these concepts, we come to sequential capsid assembly. So we've talked about the components of this, an assembly line concept, making the proteins individually or as precursors. Now, when we make the particle, there are 2 concepts that we're going to talk about. And the first one is sequential capsid assembly. Many viruses do this. This is poliovirus as an example. So here we have a poliovirus infecting a cell. The RNA is released early into the cytosol so it can be translated into proteins. And this genome is made into a polyprotein. I showed you on the previous slide. The capsid proteins are made into a polyprotein precursor. The whole genome is actually translated as a single polyprotein and then cut up. But here's the capsid protein structural unit, VP1, 2, and 3. 5 of these assemble to form pentamers. And then the pentamers assemble to form a virus. So sequential series of steps to form the virus particle. You make the 5S structural unit, you make a pentamer, and then you make the virus particle. These pentamers, by the way, associate with the viral RNA that's produced by RNA synthesis, of course, and then the RNA gets put into these empty capsids. So we think the RNA binds the pentamers then assemble into a virus particle. You need 12 pentamers to do that. And then there's a final cleavage of one of the capsid proteins that gives you the infectious viruses. But the point here is that it's a sequence of events. You do step 1, step 2, and step 3. And here's another example which I want to give you because it's very cool. It's different. The one we just talked about is polio. It happens in the cytoplasm. This is herpes virus, which is assembled in the nucleus because herpes viral DNA is replicated in the nucleus. And so the capsid proteins are imported into the nucleus and the capsids are built there. So here's a nucleus of a cell. Here are the nuclear pores. And so our DNA is in the nucleus. And all of these capsid protein components have to be made in the cytosol and imported through the nuclear pore complex. We have a portal down here. We have some protease precursors, as you'll see, and we have hexamers and pentamers that go to make up the capsid. So basically what happens here is that the capsid is assembled in the nucleus without any DNA initially. And it's so big that an internal scaffold is built. And so here we have labeled protein scaffold. All these proteins inside here are the scaffold. The icosahedral shell is made up of pentamers and hexamers, but they're held in place by the scaffold. And once this is built, once the scaffold is complete, that activates one of these proteins, which happens to be a protease, and the protease chews up the scaffold, and then the DNA is put inside to replace the scaffold. So you build the scaffold inside the building, then you take the scaffold down and you put the DNA in. It's a very clever way to do that. And how the DNA gets in, of course, is through that single portal, and we're going to look at that specifically in a moment. All right, so that's sequential assembly. 2 examples, poliovirus and herpes simplex virus. The other kind of assembly is called concerted. Just think of a concert where everything is working together. All the instruments are working together. That's what we're talking about here. Assembly happens all at once. So the genome and the particle come together. And that's illustrated here with influenza virus. Now, certainly there are parts of this that are sequential. For example, in the nucleus, we are reproducing the viral RNAs and wrapping them up in nucleocapsid protein in a series of steps. Those nucleocapsid proteins, remember there are 8 segments for influenza virus. So all 8 of those get made in the nucleus. They're exported out of the nucleus. They go to the plasma membrane where the hemagglutinin and the neuraminidase have already been inserted. Those are put there by the secretory pathway from the ER. And these RNAs know where to go. I'm going to tell you how they know that in a bit. And then as soon as the RNP hits the plasma membrane, the particle forms. That's the concerted part. The particle forms in one step by the RNPs touching the plasma membrane, and then the particle forms by what we call budding and then pinches off from the cell. So it's different from poliovirus where it's done in a series of steps, right? This is all happening. The assembly only happens in association with the genome. Remember, with polio, you make a pentamer and you put the RNA on it and then you encapsulate it. So that's concerted. Now, the proteins that are in the plasma membrane there, we've talked a little bit about them. We're going to talk a lot about them subsequently. When you get vaccinated for influenza virus, you're making largely an immune response against those glycoproteins, the hemagglutinin, and to a lesser extent, the neuraminidase. Let's look at the hemagglutinin, because it's very interesting how it gets up there and how it's processed. So again, the virus particle has hemagglutinin and neuraminidase spikes. The hemagglutinin is this club-shaped purple spike there. And that forms a trimer on the right here that we've talked about in terms of binding the receptor and catalyzing fusion so that the RNA can get out of the particle into the cell. On the bottom is a line diagram of the hemagglutinin. And it's inserted into the plasma membrane. Well, this would be the viral membrane, let's say. There's the carboxy terminus here, would be in the interior of the particle. And then the red part is the extra viral part of the HA. And you can see a number of things that are important. First, at the very end terminus, there's a signal sequence. That, of course, directs the protein to go into the ER when it's translated. That gets cleaved off eventually by a cellular protein, the signal peptidase. So the mature virus doesn't have this signal sequence. And then you can see lots of disulfide bonds. And these cause the protein to form its secondary and tertiary structures. But most importantly, down here near the membrane, you can see in green here is what I've labeled the fusion peptide. Now remember, the fusion peptide is what is hidden by the viral membrane. It's down here near the viral membrane. And then when the pH of the endosome drops, the fusion peptide is thrust up and it catalyzes fusion with the endosome. That fusion peptide has to be exposed in order for that to happen. When the HA is made, this fusion peptide is buried here. It has to be cleaved. This little arrowhead here is a cleavage site for a protease. It's a cell protease that cleaves the HA and allows it to undergo that conformational change. If it's not cleaved, the virus is not going to be infectious. But I wanted to show you where it's located and the fact that cleavage does not release the other part of the HA because it's held in place by this big disulfide here. So you maintain the two cleavage parts, which are called HA1 and HA2, because of the disulfide bond. It's very important for that. This HA undergoes typical transport from the ER to the plasma membrane. So the protein is made in the ER on ribosomes that are bound to the ER. The protein goes into the lumen in the ER where it folds. Here's a diagram of that. And then, of course, these proteins move through the Golgi apparatus all the way up to the plasma membrane, as I showed you before. And what happens in the Golgi? Some very important things like adding sugars to the protein. This is a glycoprotein, so many different glycoproteins are added. And for some influenza viruses, the HA can be cleaved in the trans-Golgi network. Remember, the HA has to be cleaved for infectivity. Most human influenza viruses, the HA is cleaved when the virus comes out of the cell in the respiratory tract. And the protease that does that is restricted to the respiratory tract. And that is probably what restricts influenza viruses to your respiratory system. The protease isn't elsewhere. You've probably heard about avian H5N1 influenza, right? It's killing a lot of birds. That's why eggs cost so much, because it's killing a lot of birds. Have you heard of it or no? Okay, good. So H5N1 influenza virus, the protease, is in the trans-Golgi. And it's in most cells, which is why that virus is very virulent, because it can reproduce outside of the respiratory tract. So we will come back to that later. But these things that we learn can be very important for understanding how viruses cause disease. Our first question for today: subassemblies are involved in which of the following types of virus particle production? Concerted assembly, sequential assembly, assembly lines, chaperone-assisted assembly, all of the above. So all of the above. Subassemblies are involved in all of them, right? Concerted can have subassemblies, sequential can have subs, assembly lines can have subs, so can chaperone-assisted assembly. It's all of the above. So let's now talk about getting that genome into the particle. This is what we call genome packaging. It's another phrase I find many people are not familiar with. What are you talking about? What I mean is putting specifically the RNA or the DNA into the virus particle. And the problem here is, of course, you have to distinguish somehow viral genomes from cellular, either DNA or RNA, because those molecules are going to be present. And the solution is what we call packaging signals in the viral genome. So please remember that. The packaging signal is a sequence in the genome. It can be RNA or DNA that is needed for the nucleic acid to be packaged into the virus particle. So let's see how this works. These are some DNA packaging signals. So adenovirus on the top, SV40 on the bottom. So adenovirus, here's the left end of the genome. And there's one of the origins of replication. And you may remember there's an early region 1A promoter here very close to the left end. This is where these packaging sequences are located. These blue bars are all packaging signals that have been identified that are needed to get this DNA into the capsid. It's a set of repeated sequences, and what they do is they bind a viral protein called 4A2, and here we have on the bottom an empty adenovirus capsid. So very much like herpes simplex virus, the adenovirus capsid is made empty in the nucleus, and then the DNA is put into it. And the way the DNA goes in is the DNA packaging sequences bind 4A2, and then 4A2 is brought into the capsid. So cellular DNAs will not have these packaging sequences in them, and therefore they won't be incorporated. And this is a very important finding because we are, as you will see in the last lecture of this course, we use adenoviruses for gene therapy. We give them to people for various purposes. And you can take those packaging sequences and put them in another DNA, and it will get packaged into an adenovirus particle. So that's the beauty of these discrete packaging sequences. You could take this piece of DNA and put it in front of some gene that you want to be packaged into adenovirus, it'll get packaged. So that's why it's important to know these things. On the bottom is the SV40 packaging signal. Again, it's a series of repeated sequences, those vertical bars. And that's just to orient you. Here's the origin of replication, right? The part where DNA begins to go bidirectionally in either direction. So the packaging signal is just to the left of that. So that's how DNA virus packaging signals work. So here's how herpes DNA is packaged. And I show you this because it's very clever. But viruses are not clever. Evolution is clever, but it's a cool process. So remember, the herpes DNA is reproduced by rolling circle. The DNA is replicated around the circle, and off come pieces that are copied on the other strand. And eventually you get genome length double strands. You may have been wondering when we talked about this the other day, how do you get from the toilet paper to individual sheets of toilet paper, right? Because that's what we're talking about. We're going to tear them apart, basically. So the genome, the individual genomes, have at their left end packaging sequences. PAC1 and PAC2 are two essential packaging sequences that are needed for recognition by the capsid. And so the way it works is, here is a capsid, an empty capsid that we've just built in the nucleus. And remember, it has one portal. And the portal is associated with a number of viral proteins, which you see here, the red and the white and the green one. And the portal and these proteins recognize the packaging sequences on the viral DNA. And so the viral DNA, which is a long concatemer, right? Many units of genomes all linked together. That's what we mean by a concatemer. That binds to the portal via PAC1 and PAC2. Step one. Then there's a motor associated with this portal. A motor meaning it can wind the DNA, it can pull the DNA into the capsid. So that's what's happening here now. The packaging signals have bound to the portal, and now the portal motor is pulling the DNA into the interior of the capsid. And it keeps pulling until it gets to another set of packaging signals, which would be at the 5' or left end of the next genome concatemer. And then there's an endonuclease associated with the portal, which cuts the DNA. And now you have a single herpes genome encapsulated by virtue of these packaging signals binding. The motor in the portal pulls it in. And then when it reaches the second set of PACs, which would be at the end of the next genome. It cuts it, and you have a single genome in the capsid. The other aspect of this that's very amazing is that the DNA of this virus, and also many other DNA viruses, including phages, is packaged under enormous pressure, 500-600 pounds per square inch. Right? That's a lot of pressure. I mean, your tires on your car are 30 pounds per square inch, right? You put too much in, they explode. So 500 to 600 PSIs. Why is that? Well, first of all, it's amazing that this little protein can do it. And the capsid is so strong that it doesn't blow up. The reason it does it is because when the portal docks on the nuclear pore, the DNA comes shooting out, right? Under pressure. It's under pressure, and that gets it into the nucleus. So just biological machines are just amazing. Now, RNA genomes also have packaging signals. Let's look at a couple of those. This one is HIV, a retrovirus. So lower left is the left end of the retrovirus genome. There's the mRNA. So we have the primer binding site here just to kind of orient you. There's the AUG that's going to start the translation of the first polypeptide, the GAG protein. And we've expanded this little region here marked psi. Psi stands for the packaging signal. That's what we call packaging signals in RNA viruses. And for this virus, that's necessary for packaging. It's not all you need. You need some other signals as well. But it consists of a number of stem loops here. And I just remind you that this stem loop 1 forms a kissing complex with another RNA. And so that's how the two RNAs are brought into the virus particle together by the base pairing between this sequences on one molecule and the second molecule of RNA. And so we always put two RNA molecules into each retrovirus particle. So that's mediated by this kissing complex. But the uptake of the RNA into the particle requires this entire sequence here. So the way it works is that this sequence interacts with a viral protein called the nucleocapsid protein. So GAG, which is the first protein made here, consists of matrix, capsid, and nucleocapsid. And the capsid is going to form the shell around the genome. But it's the nucleocapsid that binds the RNA and pulls it into the shell. You're going to see how that works in a moment. It's very neat. The nucleocapsid is expanded here, and it has two regions that bind to RNA specifically. And this part of the nucleocapsid binds this packaging sequence right here, and that gets you into this configuration in the lower right. Two RNA molecules associated by the kissing loop binding to two nucleocapsid molecules that are part of this GAG precursor. This is a polyprotein. It's not been cleaved into its components yet. So you have matrix, which is the gray protein there. The capsid and the nucleocapsid are all up there as well. It'll be more clear in the next slide. And influenza virus is a similar situation. There are packaging sequences on the RNAs. For many years, we thought that for influenza virus, the incorporation of segments was random. So there are 8 segments in influenza virus particles. And we thought that the virus, when it was built, just grabbed 8 RNAs from the cell. And if it were random, you would get 1 in 400 infectious particles. If you do the math, if you just grab from a pool of 8 RNAs in a cell, if you just grabbed 8, or I should say a pool of replicating RNAs in the cell, if you just grab 8, 1 in 400 would be infectious. And in fact, that's about the infectivity ratio, the particle to PFU ratio of influenza virus. It turns out there are specific packaging signals, because there have to be, right? Because otherwise you'd be packaging cellular RNAs, and we don't do that. So there are packaging sequences on each segment, and this is what they look like. Here are the 8 segments of viral RNA drawn as lines, and the blue sequences at either end are conserved packaging sequences. And these packaging sequences allow the RNAs to interact with each other. So you get 8 RNAs, 8 of the right RNAs into the virus particle. And they also interact with viral proteins to get them into the particle. So remember, you have to put 1 of each 8 segment. You can't put all of the same segment, it's not going to be infectious. And that's what these sequences at the ends of the RNAs do. They ensure that you get all the 8 different sequences in. And on the left is a picture of how the budding occurs. And it turns out that the segments, the RNA segments, which are shown here as colored helices, green and red and gold and blue, they're always oriented in parallel and perpendicular to the membrane as the particle is budding. And that's because of the interactions between these RNAs, these RNA sequences among the RNAs and with viral proteins. And if you look at a section in the electron microscope, you can see that all the RNAs are aligned because you can see the cross sections of each of the 8 RNAs in these particles. And that happens because of these packaging signals. So it's not random. And finally, the last mechanism of packaging is what we call selective. And this happens with some double-stranded RNA viruses. This happens to be a bacteriophage called Phe6, which has 3 double-stranded RNA segments, small, medium, and large. So this is a virus, again, with 2 capsids, and it displays a serial dependence of packaging. So the first RNA to go in, it has a small, a medium, and a large RNA, so named because they're small, medium, and large. The first one to go in is always the small RNA. It has sequences on it that ensures that it goes in first. And then the next one, the M, only goes in if S is present. And then finally, the L goes in only if M is present. So it's a serial dependence of packaging. And this apparently works really well because the particle to PFU ratio of this virus is 1, which means that almost every particle built in an infected cell is infectious. So this idea of this serial packaging probably gives you a lot of particles with the right number of segments and not some with the wrong numbers, for example. Packaging signals on viral blank interact with viral blank during assembly. Okay, so what are the blanks? We have lipids and proteins. We have proteins and subassemblies. We have genomes and proteins. We have proteases and membranes, or proteins and genomes. So which signals on viral X interact with viral X during assembly? Okay, it's genomes and proteins. So packaging signal is on the viral genome, and that interacts with viral proteins during virus assembly. Packaging signal is always on the genome. That's how we do the nomenclature. You just have to remember that. And finally, some particles acquire envelopes. Let's talk about that process. Most particles, you assemble the internal structures and then it buds out of the cell in a process we'll look at. Most envelope viruses do that. But for some retroviruses, the particle buds, it's not yet assembled. The components inside are not yet assembled. So they assemble either simultaneous with or after budding. And what drives budding? Well, different viral proteins can drive budding. So what I mean is what makes the bud occur. And you see there are 4 different ways this can happen. For example, for some viruses, class 1, the envelope glycoprotein and the capsid are needed for budding. So you have to have both of those. Sometimes it's just a matrix protein. So the matrix protein is a protein that's just beneath the envelope in envelope viruses. It gives structure to the envelope because the envelope alone would be too flimsy to exist. So the matrix protein, that blue protein there, is just under it. And sometimes if you make just the matrix protein in a cell without anything else, it will make particles. It will force the budding to happen. Sometimes the envelope proteins drive budding. This is really cool because we make vaccines actually by producing HA protein of influenza virus either in insect cells. So that's licensed. If you ever got a flu vaccine called Flu Block, that's made just the HA in insect cells. And it makes particles and they inject that into you. And that's also being looked at in plants. You can also make flu, we call these virus-like particles because there's no genome in them. So just the HA will do that. And finally, sometimes the matrix and additional proteins are needed for driving budding.
Would that be an edible vaccine?
Would a plant one be an edible vaccine? I always dreamed that you could do it in bananas and just give people bananas with vaccines in them. But no, what they do is they crush the leaves and take the sap and purify the particles. Because a lot of people have allergies to plant components, so you want to have those in there. But I think the banana one would be great. Back in the day when a lot of people smoked, I thought we could do it with tobacco. And you could smoke and get your vaccine at the same time. I think we could get around a lot of anti-vax sentiment by doing it. Because who doesn't like a banana? All right, so this is an example of budding now. Let's look at the process for a couple of different viruses. This is influenza virus, where the particle is assembled. Remember, we call this concerted assembly, where the particle forms all at once. So we make the components, we make the RNPs, the influenza RNA and proteins, and they go to the plasma membrane where they interact with the HA, and that drives the budding. And budding is this process where a little piece of the membrane begins to evaginate, gets bigger, and eventually pinches off. The pinching is done by a cellular protein, which we're going to look at in a moment. So that's a cellular process, this idea that a piece of membrane can come off. So there are signals in viral proteins to make sure that the RNPs, for example, get to the plasma membrane. Because remember, in this slide, these RNPs come out of the nucleus and they find the HA at the plasma membrane. How do they do that? So here's a line diagram of the M1 protein of influenza virus. So that's the M protein that lines the underside of the membrane. And it's shown here as these blue blocks. And eventually those blue blocks will line the inner surface of the membrane there. So this is M1. It has a number of regions that are important. First of all, it has a nuclear localization signal. So it's made in the cytosol, gets shipped into the nucleus. And then it's going to bind the RNP in the nucleus. Let's go back to the previous slide. So M protein is here. It's going in the nucleus. It's going to bind the RNP, and then it's going to get shipped out. So it also has a nuclear export sequence. So M1 directs its own import, and then it directs the export of the RNP. So also on M1 are a region at the N-terminus, which is required for membrane binding. This is what targets the M1 to the membrane. That's why it goes there and binds to the plasma membrane and eventually becomes part of the budding particle. And other viruses that bud in a similar way, like VSV, they have a protein called M protein as well. They have membrane binding regions as well, and regions that bind to the RNA, the RNP particles in a similar fashion. So that's how the RNPs get to the plasma membrane, by these proteins that have hydrophobic membrane targeting sequences. Let's take a look at retrovirus budding. This is really quite interesting also. So remember, we make RNA genomes in the nucleus. The provirus is integrated. It's transcribed by host Pol2. The mRNAs come out into the cytoplasm. And then some of those are translated. And among the proteins that are made, first is the GAG protein. Remember, you have a GAG protein made, and then sometimes a GAG-Pol fusion, which includes the reverse transcriptase. So there's the GAG, and there it is above it. It's got matrix, it's got capsid, it's got nucleocapsid. They're all still joined together. They're not cleaved yet. And GAG-Pol has GAG plus RT and integrase. It's also a polyprotein precursor. So sometimes the ribosome stops at GAG, sometimes it goes and makes RT and integrase. Now, the nucleocapsid part of GAG binds the RNA specifically in that packaging signal that I showed you earlier. So here is nucleocapsid. It's yellow. It's binding the RNA. There are two RNAs that are held together by that kissing complex. And this whole structure is going to go up to the membrane. And a whole bunch of these are going to line up to form the inner shell of the capsid. So we have two RNAs here, and then lots of GAG precursors. Now, every now and then, a GAG-Pol will get incorporated into the virus particle, and that brings the RT and the integrase into the particle. And what brings GAG to the membrane, I'm going to show you in a moment. That whole assembly eventually buds out, but it's not yet mature. It undergoes a final maturation step, which involves the protease in the particle cleaving the gag into its individual components, right? Because you've made this particle, you still have gag and gag pole in there. You have to liberate all of those individual proteins. And that is done by one of the proteins, which is a protease. And it cleaves them all. And that's how you get the final viral structure with the capsid inside, and all the individual proteins are liberated. The RT, the integrase, capsid is now liberated. The matrix, the blue protein, is now lining the inside. But they're all brought in there by nucleocapsid binding to the RNA. So what puts GAG to the membrane? That's shown here. Here's GAG again. Matrix, capsid, nucleocapsid. Matrix is expanded below here. Matrix has some sequences important for membrane binding, but more importantly, it has a sequence at its N-terminus for the addition of a lipid called myristate. It's a fatty acid that's covalently linked to the matrix protein, and it targets matrix to the plasma membrane. That's how matrix knows to go to the plasma membrane. Now remember, matrix is part of the GAG protein, so it's dragging along with it capsid and nucleocapsid, of course, is dragging along with it RNA. So all of that goes up to the plasma membrane and drives the budding of the new particles. This kind of lipid addition we see in other viral proteins. It's a common way to target proteins to a membrane without having, say, a signal sequence, which is typically the way it's done in the endoplasmic reticulum. So here's myristate. It's a fatty acid, and it is linked to protein at this specific sequence. 3 amino acids, serine, threonine, alanine, any amino acid or C, that's the signal for the addition of myristate. And so you make the protein in the cytoplasm, and then you add the lipid, and it will go to a membrane. Otherwise, you would have to put the protein in the ER and get it transported to the plasma membrane. It's a little more involved. And these are internal proteins. They're not transmembrane proteins. So myristate allows you to target proteins to membrane. So if you change these amino acids, for example, and you made a virus that did not have myristate linked to M protein, you wouldn't make any particles, because you need to bring M protein to the plasma membrane along with all the other proteins by virtue of this myristate. Finally, let's talk about how the bud forms. We talked about this process of budding. How does that happen? So the GAG protein, again, shown up here for 2 different retroviruses, HIV and a murine leukemia virus. We have matrix, we have capsid, nucleocapsid, matrix, capsid, nucleocapsid. But you see there's some extra proteins in here I haven't talked about. There's P6 and P12. And so years ago, people looked at these and said, what are these doing? And you know what we do in virology when we want to know what a gene or a protein does? We make changes to it. We take it out or we make amino acid changes. And what people found when they made changes to P6 or to P12 is that you would get virus budding initiated, but it would never finish. You would get these particles stuck on the cell by these short membrane tethers. Right? So here's the cell. It's making virus particles. And you can see they're all budded off, but they're never released. And that happens when you remove this P6 or the P12 sequence. So these were called late domains because of that, because they affect a late stage in reproduction, right? The budding is about as late as you can get in the infectious cycle. And then people did more work. They found these sequences in a lot of other viruses that bud from the membrane, a lot of plus and minus strand envelope viruses. And it turns out that they interact. These proteins, P6 or P12, late proteins, they interact with a series of cellular proteins that are part of what we call the Escort Pathway. This is a pathway involved in membrane scission events. And these are all different protein components, which you absolutely don't need to know about, but I'm showing them to you because, for example, P6 interacts with these components of the Escort pathway, and P12 interacts with this one. So basically what is happening is we have a cellular process, a cellular machinery, if you will, which has evolved to pinch off membranes. So when cells divide, the division of the two cells is called abscission. That's carried out by this Escort pathway, which stands for endosomal sorting complexes required for transport. So here is one of the components of Escort. It's mediating the pinching of the two membranes apart. It's a very important pathway. Sealing of the nuclear envelope at mitosis is done by this pathway. Making of exosomes. You may know exosomes are vesicles made in cells that can carry RNAs and proteins. They come out, fuse with the plasma membrane, and go to other cells. That's controlled by the Escort pathway. Multivesicular bodies. So what viruses have done is to, I don't want to say steal because that would make them do a human thing. They've evolved to require components of the Escort pathway. And they steal them by binding to these P6 and P12 proteins. So the P6 and P12 proteins of the retrovirus, right, they're part of this growing bud here. They recruit the Escort machinery, and then once the bud is formed, the bud is driven by viral proteins. But to get it pinched off, you need this Pac-Man thing here, right? VSP4, part of the Escort machinery. To pinch it off, that's the final step. So if you make, if you delete the P12 and the P6 proteins, you get viruses stuck by a stalk. So you don't need Escort to make buds. You need Escort to pinch it off. So the viral proteins recruit this pathway to the bud, and that's how they leave the cell. It's quite amazing, right? Now, glycoproteins, as we've talked about so far, going to the plasma membrane, but they can also go to internal membranes. And here's a schematic of different viruses whose glycoproteins are going in various places. Look, herpes simplex viruses can put their glycoproteins in the nuclear membrane. Coronaviruses put them in the ER, as do some other viruses, vaccinia, flaviviruses. Bunya and vaccinia viruses put them in the Golgi apparatus. And of course, many viruses put their proteins in the plasma membrane as well. But the point here is that viruses can bud into various cellular compartments. So here's an example for coronaviruses. So here's a cell that's reproducing a coronavirus. We're making mRNAs, making viral proteins. Here are the viral glycoproteins made in the ER. They get transported to the ER Golgi intermediate compartment. Excuse me. Here, the genome is being replicated. It's coated with nucleocapsid protein. And then the nucleocapsid buds into the ergic. So it's budding into an internal vesicle. It's not budding from the surface. And of course, this budding requires the Escort pathway. This vesicle then, containing a complete coronavirus, moves to the cell surface. The outer membrane fuses with the plasma membrane, and that releases the particles. So coronas do not bud from the plasma membrane. They bud into the ergic, and then they're transported up to the cell surface. Just so that you know, there are different ways to achieve the same end. All right, our last question is, which statement about budding, viral budding, is incorrect? A, the envelope can be acquired after or simultaneous with assembly of internal components. B, the viral spike glycoprotein can drive budding. C, no host proteins are involved in budding. D, lipids assist structural proteins to interact with the membrane. Or E, the viral membrane can be acquired from the nucleus, ER, Golgi, or plasma membrane. Which is wrong? So what's wrong is that no host proteins are involved in budding. Remember the Escort proteins. That's a cellular system of pinching off the bud. Everything else is correct. The envelope can be acquired after or simultaneous with assembly. Spikes can drive budding. Lipids assist proteins to get to the membrane, like myristylation of M. And we can get the membrane from pretty much anywhere. And speaking of getting the membrane from anywhere, I wanted to show you a schematic of herpes. You know, herpes needs to make everything complicated. I guess that's par for the course for a virus that's with you forever. So herpes DNA, right, is reproduced in the nucleus. The capsid is formed in the nucleus. It then buds out of the nucleus to acquire a membrane. So now we have a nucleocapsid with the DNA. We have a membrane around it. And how does it get out of the ER? It fuses with the ER membrane, and the nucleocapsid ends up in the cytoplasm. So it's lost its membrane. Not an efficient process. So what does it do? Well, it acquires another membrane by budding into the trans-Golgi network, part of the Golgi. So now in the Golgi, we have herpes virions with membranes, and they don't fuse with the Golgi, fortunately, otherwise they would lose it again. Although you could imagine the capsid coming out and then acquiring a membrane at the plasma membrane. That should work, right? But that's not how it happens. These viruses bud off of the Golgian vesicles. Vesicles go up to the surface, they fuse, and out come the virus particles. And these are electron micrographs of each of these steps. The capsids forming in the nucleus, the capsids budding from the nuclear membrane into the ER, and so forth, all the way up to free virions at the surface. So this virus acquires a nuclear membrane, loses it, and so the membrane that's on the virus particle is derived from the trans-Golgi network. Many other things happen on the way from the ER to the plasma membrane that are important for virus biology. So this is an example of a flavivirus, one of these envelope viruses where the glycoproteins are flat on the surface of the particle. And I want to illustrate the changes that occur as this virus moves from the ER to the plasma membrane. So the viruses bud into the ER. They acquire their membrane at the ER. And then they're transported through the vesicular transport pathway through the trans-Golgi network where the pH drops. So the pH goes from 7 to 6.7 to 6 and to 5.7. And then these viruses are released by the vesicular transport pathway. This is actually an envelope virus, but the surface is covered with glycoproteins, so you can't really see the envelope there. And of course, the extracellular milieu is more neutral. What is happening here is that the glycoproteins on the mature particle have to be flat. They have to be spring-loaded in order for them to catalyze fusion when they enter a new cell and the pH drops in the endosome. So they have to be spring-loaded, which means they have to be flat on the surface. And these little red colored areas here, those are the fusion peptides. Those have to be able to be flipped up and put into the membrane. As the virus is assembled initially in the ER, these glycoproteins are pointed up, and the fusion proteins are covered with this blue protein that protects the fusion protein so it doesn't fuse in the low pH of the trans-Golgi. So that's a protector, which remains with the particle until it's released from the cell. So the low pH of the trans-Golgi pushes the glycoproteins flat on the surface. It spring loads them. There's a protease in the trans-Golgi, furin, that cleaves this blue protector from the fusion peptide. And then when the pH is fully neutral, then that blue protein comes off, exposing the fusion protein. So now in a new cell, as the virus enters, as the pH drops, the proteins will flip up and the fusion peptide will be exposed for insertion in the membrane. Okay, so this has to be spring-loaded in the trans-Golgi without causing fusion to happen, and that's what that blue protein does. So it's a really interesting series of events. I want to end up just by briefly talking about how viruses leave cells. We've talked about how viruses leave by budding. They can leave, this is an epithelial sheet in your respiratory or gastrointestinal tract. Viruses can leave by the apical part of the cell. They can also travel from cell to cell. As we'll see, they can also be released from the basolateral domain of the cell and enter your circulation. But some interesting tricks have evolved. For example, cells infected with vaccinia virus, they induce a structure that propels the virus from one cell to another. So here's a vaccinia virus infected cell. The mature virus is transported to the surface by motor proteins on microtubules. It actually has two membranes. So the outer membrane fuses, so you have this extracellular virus particle, which interacts with a receptor on that same cell. This X is the receptor, and triggers actin polymerization. Actin, of course, forms the microfilaments below the plasma membrane. And what this actin polymerization does is pushes the virus into a neighboring cell. You can see the virus is sitting here on the cell surface. The polymers of actin push it into a neighboring cell, facilitating its spread from cell to cell. So here's a fluorescence micrograph of an infected cell where these green fibers are the actin filament-driven extensions, and the vaccinia virus is the red at the tip. And there's an electron micrograph of one of these actin filaments with a vaccinia virus at the tip. So it's an interesting way of ensuring that the virus gets to another cell, which of course it has to do. Now for viruses without membranes, how do they get out of cells? Like poliovirus. Well, there are two general ways. One is that the viruses kill the cell. They break the cell open and out comes the virus particle. So I showed you this time course of poliovirus infection of cells early on. The cells are rounding up, they're coming off the plastic, and eventually they break. And so cell lysis can be caused by programmed cell death like apoptosis and necroptosis. Many viral proteins have been identified that rupture cell membranes and allow virus particles to come out. In fact, some of these are called viral porins. They poke holes in cell membranes. And as viruses also inhibit cell protein synthesis, the membranes lose integrity. They fall apart, and out come the viruses. So that's a very straightforward way that viruses can get out of cells, simply by breaking them open. But there's another more subtle way that I want to leave you with, and that is that some non-envelope viruses, like poliovirus, can get out of cells without the cells lysing. And that's because in cells infected with, say, poliovirus, remember, the virus induces the formation of double membrane vesicles because it's on the surface of those vesicles that the RNA is made. And these vesicles are essentially formed by a normal process called autophagy. And autophagic vesicles late in infection, what autophagy does is when a cell feels like it's going to die, it generates these vesicles that grab pieces of the cytoplasm and throw them outside for someone else to use. And so these are the vesicles here being formed. They capture cytosolic components and then they release them at the plasma membrane. And so it happens that viruses can get captured in these autophagosomes and released in a non-lytic way before the cell is lysed by infection. And more recently, it's also been found that in some virus-infected cells, virus particles may end up in exosomes. Exosomes are, of course, as I mentioned, small vesicles made in cells that can deliver RNA and protein to other cells as kind of a signal. These can also capture virus particles and release them, again, without the cell being lysed. So that is the end of our reproduction of virus. Next time, we're going to take a look back and we're going to try and integrate all the things that happen in an infected cell in order to facilitate virus multiplication.